Microbial Communities in Living Organisms, Cattle Rumen Bacteria, a project by Elizabeth Hyde, Caitlin Miller, and Melissa Andolina.
A ruminant is an animal with a four-chambered modified simple stomach. Examples of ruminants include cattle, goats, and sheep. The rumen is one of four chambers in the ruminant stomach. It is characterized by its papillary lining, which increases its surface area for absorption, fermentation, and anaerobic respiration. This chamber houses the symbiotic relationship between cattle and their microbial community. Symbiotic mutualism is a relationship in which both parties benefit. While bacteria within the rumen are provided with proper anaerobic living conditions and sustenance, cattle utilize the byproducts of bacterial metabolism for their own metabolic processes.
The microbial profile of the rumen includes a majority of bacteria and protozoa with a minority of yeast and fungi. The most numerous of these bacteria are Bacter succinogenes, Ruminococcus albus, Butyrovibrio fibrosolvens, Clostridium leucoheti, and Streptococcus bovis. The main function of these bacteria are to digest cellulose, xylanose, and starch, which are the major constituents of a forage diet. The major products of the bacterial digestion are volatile fatty acids such as butyric acid, propionic acid, acetic acid, and formic acid. This slide contains images of the most numerous bacteria within the rumen. The following are obligate anaerobes: Bacter succinogenes, a gram-negative rod; Ruminococcus albus, a gram-positive coccus; Butyrovibrio fibrosolvens, a gram-positive rod; and Clostridium leucoheti, a gram-negative rod. The final image on this slide, Streptococcus bovis, is a facultative anaerobe. It is also a gram-positive coccus.
The rumen microbial demographics are established postnatally through feed management. Microbes divide themselves into three strata based on oxygen availability and type of feed digestion. A portion of the bacterial population are facultative anaerobes, meaning both aerobic and anaerobic methods of energy production can be used. Under anaerobic conditions, these organisms can utilize many different electron acceptors, such as iron, manganese, nitrate, and nitrite in replacement of oxygen as the final electron acceptor. Similarly, obligate anaerobes possess the methods of energy production without oxygen as an electron acceptor. The two main groups of bacteria housed within the rumen are cellulolytic bacteria, which break down fibers and cellulose provided by forages in the diet, and amylolytic bacteria, which digest starch provided by grains in the diet.
Anaerobic respiration does not use oxygen in this energy-generating sequence. The final electron acceptor is not oxygen following the electron transport chain. Anaerobic respiration uses alternative inorganic compounds as the final electron acceptor. Anaerobic respiration begins with glycolysis. Glucose, with the use of ATP, is transformed to glucose 6-phosphate, which then transforms to fructose 6-phosphate. Again, with the use of ATP to ADP, it transforms to fructose 1,6-biphosphate. It then is transformed into dihydroxyl acetone phosphate and glyceraldehyde 3-phosphate. Dihydroxyl acetone phosphate is then transformed to glycerol aldehyde 3-phosphate, which is then continually transformed with the use of NAD plus to NADH, a reduction reaction, to 1,3-biphosphoglyceric acid. 1,3-biphosphoglyceric acid is then transformed into pyruvic acid through the reduction of 2 NAD plus to 2 NADH, and 2 molecules of ATP are produced. Pyruvic acid then enters the Krebs cycle. It is transformed into oxyacetic acid, and the acetyl group from acetyl-CoA is donated to generate citric acid, which then transforms into isocitric acid, which is oxydecarboxylated via the reduction reaction of NAD plus to NADH to form alpha-ketogluteric acid. This then undergoes another reduction reaction of NAD to NADH with the release of CO2 to create succinyl-CoA is hydrolyzed. With this additional energy, ATP is formed. This reaction releases CoA to make succinic acid. FAD is transformed to FADH2 through oxidation to form fumaric acid, which is then hydrated to malic acid, and NAD is reduced to NADH to form oxaloacetic acid. Electrons from glycolysis and the Krebs cycle are carried via FADH2 and NADH into the electron transport chain. Here, electrons from NADH are carried to protein complexes containing flavoproteins and iron or sulfur, which rotate between oxidative and reductive states to carry electrons. These electrons travel via ubiquitinones and cytochromes, which are shuttles between structures. Cytochromes are membrane-bound proteins which contain a heme group to carry electrons. Electrons then enter ATP synthase, a protein channel which protons diffuse from outside into the cell. NADH passes electrons and hydrogens to protein complexes at the start of the electron transport chain. These hydrogen protons are pumped out of the cell. FADH2 also passes electrons to the electron transport chain, and these protons are also pumped out of the cell. The protons are pumped back into the cell through ATP synthase to catalyze ATP formation. The final electron receptor of anaerobic respiration are inorganic compounds other than oxygen. During fermentation, you still carry out glycolysis to produce 2-pyruvate, which are further fermented to generate various byproducts the ruminant can use.
Clostridium fermentation produces butyrate. Streptococcus fermentation produces lactate. Propionibacterium fermentation produces propionate, acetate, and carbon dioxide. The 3 volatile fatty acids are used by the ruminant in various functions. Acetate is transported straight to the tissues to be oxidized to create ATP. It is also the main source of acetyl-CoA for synthesis of lipids. Propionate is broken down by the liver to produce glucose through gluconeogenesis. Butyrate is removed from the rumen as the ketone beta-hydroxybutyric acid and is oxidized by tissues for energy production. Carbon dioxide is erectated up the esophagus via belching.
Another product of fermentation is methane gas, produced by methanogens or obligate anaerobes in the domain archaea. Through methanogenesis, a form of anaerobic respiration, hydrogen is the electron donor and carbon dioxide is the electron receptor. Methanogens can also utilize other chlorine compounds for methanogenesis.
Holy cow! Pictured here is NC State's Hoover. Hoover is a fistulated bull. A cannula is placed in his side to allow direct access to his rumen. Professionals use these devices to collect samples during dietary trials to generate microbial profiles and grade feed efficiency. Cannulas allow observation of digestive processes and serve as a major research entity. Cattle's microbial communities are essential to the health and wellness of the animal. One practical application of fistulated cattle is transfaunation. When the rumen's microbial population is unbalanced or extinguished, rumen fluid is transferred from a healthy animal to repopulate the rumen with bacteria. Microbial depletion can occur due to rumen acidosis, being off feed for long periods of time, or being treated with antibiotics. A secondary practical application is to use fistulated cattle as a herd management technique. Typically, one herd member will have a fistula to take routine samples to allow farmers to evaluate feed efficiency of the herd. We would like to thank you for watching our presentation. We hope you've enjoyed it.